It's okay. Thank you very much, Stefan, for this nice introduction, and it's really a pleasure to be allowed to present to you on this last day of the conference the work in my group. And as Stefan already said, we are currently at the Department of Bio-Nanoscience. It's a new young department that exists since 5 years, where we combine physics and biology at the University of Delft, the Technical University of Delft. So this is where the logos refer to at the top of the slide here. This Department of Bio-Nanoscience is also part of the Kavli Institute for Nanoscience in Delft. But I should have maybe put also the logo of my previous employer, which is the FOM Institute AMOF, because necessarily a lot of what I will talk to you and present to you today was done when my group was at this institute in Amsterdam. So what I will focus on today is minimal systems for the study of microtubule-based cell polarity in fission yeast, and it's work in progress. It's not a complete finished story yet, but it's a pleasure for me to present it here to you, just to get some feedback and ideas about how to think about this project. And this project is done by a former postdoc, Pierre Couvreux, who just moved to the University of Marseille a while ago, and a PhD student, Nouria Taberner, who is finishing her PhD at the end of this year, together with a postdoc, with the help of a postdoc, Sophie Roth, who just moved to a position in Brussels. And the other names you see here on the slide are other PhD students, postdocs and technicians who are currently in the lab. So very importantly, this work was done to some extent with some theoretical work as well. I will show just one slide of it, but this is done together with PhD student Thomas Sokolowski, or I should say a previous PhD student from Pieter Reint and Wolder's group at AMOF, which is where I was before. So let me take one step back and just introduce to you this topic that, as Stefan said, that I've been interested in for a long, long time, which is, I will get back to that movie later, which is the physical, biophysical aspects of dynamic microtubules. So what I show you here is 3 examples of processes that happen in cells where the dynamic properties, the fact that microtubules grow and shrink, and also the forces that these growing and shrinking polymers generate, play an important role. So the one on the left is, of course, a famous example of a dividing cell. This movie, I think, is taken from a microscope manufacturer's website, where you see in green the microtubule cytoskeleton forming the mitotic spindle, which is responsible for putting the chromosomes, which are stained here in red, first in the middle of the cell, and then separating them into 2 new daughter cells. Here you see an analogous process in a first cell stage embryo of a C. elegans. This is a picture taken by Stefan Griel when he was in Tony Hyman's lab as a PhD student, where you also see, in this case, you do not see the chromosomes, but you see very nicely how this mitotic spindle is organizing with itself within the confined space of the cell, and also positioning itself precisely with respect to the anterior-posterior axis of this embryo. So microtubule dynamics are forces generated by these dynamic microtubes are really essential for making mitosis work properly, and particularly what we are interested to position the spindle properly with respect to the cell shape and size. So another topic that we're interested in, and which is the focus of today, is how the same microtubule cytoskeleton in a different cell, in a different circumstance, in this case in an interphase cell that of a fission yeast that is just growing, is responsible for defining where the growth zones are. So this microtubule cytoskeleton in this case is responsible for bringing cell end markers, shown here in green, it's a molecule called T1, to the cell ends, and it's this T1 which subsequently recruits the actin-based machinery that allows these cells to grow only at the cell ends and not somewhere at the side. And then the last example where, and again, microtubule dynamics, of course, are very important, not only to bring these molecules here to the cell end, but also to keep the nucleus to which these polymers are attached positioned properly within the cell shape. So a third topic, or a third example, is shown in this movie here. It's an interest that something that we got interested in recently. This is a movie from the lab of Elaine Fuchs, where you see in blue, you see actin cables, stress fibers that are hooking up to focal adhesion points, which are these pink dots in a motile cell. And what you see in green are not the entire microtubules in this case, but just see an Ntracking protein. So this is a protein that travels at the growing ends of microtubules specifically, and these growing microtubule ends, they are guided along the actin filaments towards the focal adhesions, where they help to control the turnover of these focal adhesions. So if you disrupt this interaction between microtubule tips and the actin cables, microtubule ends do not end up the focal adhesions, and the focal adhesion sites are enlarged. So this shows that in cells, very often, the microtubule and actin cytoskeleton work together, and we've recently also became interested in trying to understand to some extent how these two cytoskeletal systems influence each other's organization. Now, we do not work, except for this exception here, on these in vivo systems. Instead, the way we approach these problems is to build very simplified in vitro reconstituted artificial systems, if you like, and then ask if we can understand some of the mechanics, the biophysics that goes on in these different processes bottom up. So we take a minimal number of components, sometimes putting things in confined spaces, and then ask if you have just this limited number of components with the dynamics and the forces that it generates, can we understand at least the basic principles of how these processes happen in cells? So one example for the mitotic spindle, and I will spend some time on this at the beginning of my talk, is that we use microfluidic droplets or microfabricated chambers to study the positioning of microtubule esters or even semi-spindles with a minimal number of components, and really try to understand what is the balance of forces generated by these microtubule systems that lead to this nice positioning of a double ester in this case. In the case of the polarity of the fission yeast, we use a similar in vitro approach, which is what I will show today, but we also use a minimalistic in vivo approach where we try to reduce the natural complex of molecules, the complex network of molecules that leads to polarization of these molecules to a much simpler mechanism, and see how far we can get bottom-up in vivo. And then, but I will not talk about this, we also reconstituted the interaction with actin and microtubules in an vitro experiment. So I'm showing you all this just to emphasize that what are our approaches. So we use microfabrication, microfluidic techniques, purified proteins to really build up these systems. And then, of course, we necessarily do not get to the full complexity of a real cell, and we will never tell you the complete story. But at least using this approach, we see what are the simplest mechanisms, the simplest principles of the mechanical aspects of these biological processes. So, as promised, I will spend a little bit of time on our work on force generation and how this contributes to positioning of spindles in confined spaces. This is all published work, but I want to show it also because it introduces some of the technologies and the methods that we use. So, just a step back, so microtubules are dynamic polymers. This is again from a textbook picture where you see that individual microtubules are, of course, hollow cylinders that grow by assembling tubulin dimers at their end, which are associated with GTP, and this leads to growth of these filaments, which you can also see very nicely in these yeast cells. And every now and then, due to hydrolysis of the GTP at the microtubule ends, there's a switch to a shrinking state where GDP tubulin is released, and then the filament rapidly disassembles, which you can also see very nicely in this movie, where you see individual polymers reaching the cell ends, then switching to a shrinking state, and shrinking again. And both the growth and shrinkage of these filaments can generate forces. So the growth of a filament, since this filament is very stiff, can generate a pushing force, and this you see very nicely also in fission yeast, where when a growing filament hits the boundary, it actually bends the filament to some extent, which is a signature that a pushing force is generated. But also the shrinkage of these filaments can generate pulling forces, and this is not as intuitive to understand, because then you somehow have to imagine that the shrinkage of the filament, which happens here, has to be hooked up to something that can dynamically stay attached to the shrinking filament and thereby drag something along. But this is actually what happens, for example, to chromosomes during anaphase when they are pulled to the two new daughter cells by shrinking filaments. But it's also believed to play a role in the positioning of, for example, this spindle in the C. elegans embryo, where shrinking microtubules associate with molecular motors here at the periphery, and this allows pulling forces to be generated at the cortex. So we have, over the years, developed several assays, and again, I will just show you the pictures and not the data, is to really show that these single filaments, without anything else, can generate pushing and pulling forces and measure them. So we've used microfabrication techniques to make glass barriers as an artificial cell boundary or whatever, and then let individual microtubules growing from a stabilized seed run into these barriers, and just as in the fission yeast, you see that when they grow against these stiff barriers, they bend, and from the bending properties and from a quantitative analysis of this bending, we can measure the forces that are generated by single filaments. Or alternatively, we could do the same experiment with an optical tweezer, where we don't stick this seed to the surface, but we stick it to a glass bead, and then we can trap the bead in an optical tweezer, align this nucleation site along a second line trap, and then have this filament interact with a glass barrier again, and then use the bead displacement to measure pushing forces. We can also use this technique to measure pulling forces. What we've also demonstrated by using gold barriers instead of glass barriers, which were coated with molecular motors. So remember I said in the C. elegans embryo, it's believed that the molecular motor, specifically dynein, a minus end-directed motor, can attach to dynamic microtubule ends, and that this together can mediate a pulling force, and we showed in these in vitro experiments that that's all you need. So you just have to put an active dynein molecular motor, or multiple of them, on a barrier, and then a shrinking filament, which is hooked up to a bead on the other side in an optical tweezer, will be pulled towards the barrier. So simple microtubules just combined with molecular motors, dynein attached to a surface, can generate pushing and pulling forces. And what we then can also do is show how this simple combination of pushing and pulling forces, again in the absence of anything else, can help microtubule esters to position themselves. So the first experiments we did a long time ago were done in square chambers, where we take this is an electron micrograph of like a swimming pool dug into a glass surface. The only thing is that we put a little bit of gold on the edge, so it's really a swimming pool dug into a sandwich of glass, gold, and glass. And then we can use, again, gold-specific chemistry to put molecular motors on the edges of these chambers, sort of making a 2D cell with motors on the periphery, and showed early on that this procedure leaves the motors actually active on the side. So this is a microtubule that is stabilized, which is doing a gliding assay around the corner of one of these chambers. Now, if you put a microtubule ester and you then put dynein molecular motors on the edge, or you leave them out, you can show that both pushing and pulling forces can lead to very faithful positioning of a single ester in such a chamber. And whether the pulling, as in this case, is important to contribute to precise positioning or not, or whether pushing by itself can also lead you to the center, depends on a variety of parameters. So this particular picture show that if you have only pushing forces and the microtubules become very long, they will eventually bend and buckle and destabilize this central position, whereas with pulling forces, you always put this thing exactly in the middle of the cell. But this is not a general message. If the microtubules are shorter, for example, or if the geometry is elongated, then pushing is just as efficient a mechanism as pulling is in this case. Now, the last type of technique that we use in these experiments is based on microfluidics. So this is something that we did not invent, of course. We actually learned how to do it from Patrick Tablin's group in Paris, where we use a PDMS chip. And we push a water phase that contains buffer and proteins, in our case centrosomes and tubulin, into an oil phase through a thin restriction, and out come little droplets of water surrounded by oil and with a monolayer of lipids at the interface. And these droplets are nice because they're now 3D, they're really spherical, they're no longer quasi 2D square cells, but they're really spherical. And you can make them very faithfully of any size you want, so we make them around 20 micrometers in diameter, but they're all more or less the same size, It's a nice method because you don't use a lot of protein, you don't lose any protein, it's very gentle. Microtubules happily grow in these droplets provided that you put enough oxygen scavenger and bleaching precautions and things like this, but it's something that works really, really well. And then if you repeat the experiment that I just showed you, so we now put dynein molecular motors again on the periphery. In this case, we hook them up to the lipids, biotinylated lipids. Then we can show that if you, in this geometry, at this size of chambers, when the microtubules are long, esters do actually not position in the middle of the cell, and this results are really supported by a model that was done in collaboration with Rui Ma and Nana Pavin, who is sitting here, which sort of gave a side-by-side comparison, which was very nice between our experimental results and a mechanical model of what's going on. Instead, if you put molecular motors on the edges, both the theory and the experiments show that as long as microtubules are long enough to reach all the sides, that this ester gets very nicely pulled to the center of the spherical droplet and positions nicely in the middle. So there's a big difference between trying to put the single in the middle by pushing or by pulling forces for this specific geometry and size of the droplets. Now it's very tempting to put in 2 centrosomes and see what will happen. If you put just 2 of them, then the same thing happens. Both of them are pushed towards the edge with microtubules bent around the periphery, but they also repel each other, so there's some steric hindrance which leads to always opposite side of the droplet positioning of these 2 centrosomes. And then you can ask, now I have a repulsion between the centrosomes here. What if I put molecular motors, like in this case? Then there should be a competition between the pulling forces trying to put this thing in the middle of this chamber and the repulsion forces trying to separate the 2. And this is where you get structures that start to resemble the configuration of a mitotic spindle. So you find that the repulsion force is balanced by a centering force, and you get a steady state separation between the 2. And this, of course, is not at all, it's not a real mitotic spindle. There's loads of proteins missing that we know are very important in real spindles, but it provides us with a platform to now, if we understand the force balance that goes on in this very simple system in this confined space, what do additional force generators like molecular motors or crosslinkers add to this story, and which ones really change the force balance and which ones don't, and which ones, if you add both of them, for example, compensate each other. So it's a platform for bottom-up inquiry of the role of these other proteins. As a first example, we put in ACE1. This is a molecule that cross-links antiparallel microtubules. So it sits between the 2 centrosomes. And as you see between these 2 pictures, just adding ACE1 bundles some of the microtubules in the middle, but it doesn't really seem to change the overall configuration. Now you can also put minus and directed motors here that slide, which I didn't bring the picture, and then these centrosomes start to move apart. So you can really individually examine the effect of each additional component. Okay, so much for a long introduction. And let me now go back to the main topic of today. And like I said, this is work in progress. None of it is published. It's not even finished yet, so we'll see how it goes. So what obviously I really like studying the dynamic properties of the microtubules in a biological context, but one of the reasons I like it so much is really a multitasking system. So this, I show you again a picture or a movie of a fission yeast. This one is taken by Von Tran when he was in Fred Cheng's lab in Colombia. And in this particular movie, you see very nicely not only the microtubules, but also the nucleus and the nuclear membrane. So the microtubules are attached to the nuclear membrane. They are attached in an antiparallel fashion, such that plus ends, the growing ends, go to both cell ends. And as I told you so far, the growing filaments generate forces here, and it's the forces that are generated randomly at both ends that keep the nucleus in the middle of the cell. So even when the cell grows twice as long, the microtubules also grow longer, and the forces still balance as soon as the nucleus sits in the middle. So this system is there to keep the nucleus centered by the forces that are generated by the filaments. But at the same time, this arrangement of microtubules is important for cells to figure out where their cell ends are and where they should grow. So this I also already showed you, but you see now a similar cell, but in this case the protein T1 is highlighted, which is traveling at the tips of microtubules, so it's only traveling at the growing tips of microtubules. That's why you see these dots accumulating at cell ends, and here is a still image of both signals. And by this molecule defines the growth zones, as I explained in the middle, such that cells only grow in a linear fashion. And if you disrupt the microtubule cytoskeleton, and several people have done that in the past, you will find that these ends, microtubule ends, do not reach the poles anymore, deliver their cargo somewhere in the wrong place, and cells start growing in the wrong shape. But the same system not only puts the nucleus in the middle, defines where the growth zones are, it also enables the cell to measure how long they are. So there's another molecule called POM1, which is also localized at cell ends in a microtubule-dependent way. It's an indirect dependence, but because its localization actually depends on the localization of T1. And this activity of this molecule here suppresses the assembly, and I'm giving you the sort of overly summarized version of this story, and I apologize for the people who know much better than me how this works. But the activity of the POM1 suppresses an activity that allows a cytokinetic ring to be formed in the middle. So these two cell ends have to grow far enough apart from each other, such that the activity of this molecule in the middle becomes negligible, and a cytokinetic ring can form such that the cell divides. So now, why are we interested in? So if you go through the literature, and I'm summarizing here the work of many, many people, and there's like people like Paul Nurse, Damien Brunner, Ken Sawin, Fong Tran, Sophie Martin, who really did a lot of beautiful work figuring out, and I'm probably forgetting some people. And I'm going to sort of bluntly summarize what they've discovered in this schematic. And again, I'm missing some details for sure. And I just show it to emphasize that to understand how these molecules end up at the cell ends, how they keep this polar distribution, appears to require really a complicated regulation network. So some of the molecules, and we don't have to remember them all, that are important for making these polarity patterns work are shown here. So there's a molecule called MAL3, it's the homologue of EB, which defines where the microtubule cell ends are. Then there's a motor protein, T2, which carries cargo, including T1, but also TIP1, which is a CLIP170 homologue. And they all need each other for faithful Ntracking of the microtubule, and therefore for faithful targeting to the cell end. Then there needs to be a receptor, MOD5, which in a feedback loop by all these other molecules is also targeted to the cell ends. And then as soon as this cluster of molecules, there's another one, T4, that is important, that's there. They then trigger a phosphorylation-dephosphorylation cycle of POM1 locally that modulates the membrane affinity of this POM1, and that then causes a polarity gradient also for this POM1 molecule. So, and it's really amazing how much is known about this system and how well it's worked out. And what we were really interested in is sort of well, first of all, to try to quantitatively understand this whole network, and our way of doing that, of trying to contribute to that, is to see, okay, how can, instead of trying to understand the complete system at once, is to strip it down to its essential components and see if we can, in the spirit of our other experiments, build up a polarity mechanism also in a simple in vitro system. So that's the goal. We want to see if what of all these interactions and mechanisms is really essential for defining polarity, and if you know that, what then would be the role of all these other components that are, or what would they contribute to the system. So we do this in two ways. So the ultimate goal would be to try to reconstitute in our microfabricated chambers. We would like to make a biomimetic yeast cell, an artificial yeast, in a microfluidic droplet or in a chamber, and then confine aligned microtubules in them and see if we can put a limited number of components and establish a polarized chamber or droplet from molecules derived from this system. So that's what I will show you last, and we're not there yet. But at the same time, we also try to use the real yeast cell as a test tube, because at least it already has the right shape and size and the correct microtubule distribution, and see if we can put in much simpler molecules or molecular functionalities in this in vivo cell to also get a polarity pattern. So that's what I will show you first. So when we thought, okay, what would be, as a hypothesis, what would be the minimal functionality in this whole network that might be needed for creating a polar pattern? And we thought, okay, at least you need something that, of course, interacts with the membrane, and you need something that knows about microtubule growing ends, interacts with growing ends. Now, if you look in this schematic, even though, of course, it's just a schematic, there's no single molecule in this whole complex network that combines these functionalities. There's molecules traveling at the ends that interact with others, which then interact with molecules at the membrane. But we thought, what if we just take a chimera artificial protein that can interact with microtubule ends and has affinity for the membrane and leave everything else out? So no feedback, no phosphorylation cycle. Could that be enough to polarize a yeast cell? And so that's what we did. To make this protein, we made use of the knowledge of this system, and says that we took MAL3, which is a molecule that naturally interacts with microtubule ends, but normally travels only through the cytoplasm and is never associated with the membrane, and hooked it up to a fragment of POM1. And this is a fragment that we received from Sophie Martin's lab, and she had shown in a Cell paper that if you take this fragment, it does not polarize, it just sits homogeneously on the membrane because it cannot be phosphorylated and desphosphorylated, or it misses its autophosphorylation abilities. So if you take just this membrane anchoring fragment plus MAL1 that interacts with microtubules, we find that you can in fact already polarize cells. So this is what I showed you at the very beginning. It's not as nice, it's not as perfect, but it's these two simple molecular functionalities that allow these cells already to create a polar pattern. So these are just individual cells. We put GFP on this molecule, and this is where you see the polarity pattern. This is a movie where you see also the microtubules stained. So these are nice pictures, but we also try to quantify how good is it compared to the real polarity patterns. Is it as focused at the cell ends or not, and is it different in the dynamic properties? So just let me show you first how polarized it is. So first there's some controls. This is just a POM1 fragment without the MAL3 and microtubule. So as I promised, it's sitting homogeneously around the membrane, as also shown by Sophie Martin. And then we can take our full chimera but get rid of the microtubule, so there's now no interaction with microtubule tips, and it again sits homogeneously around the membrane. But now if you put microtubules, then the chimera all of a sudden polarizes, and you can sort of measure this. These blue lines are individual intensity profiles measured around the end of these cells, and then we averaged very many of these profiles into this red curve. And you see that there's a few microns around the cell pole where the intensity is elevated, and this range over which the intensity is enhanced is very similar to the pattern of T1 or POM1, even though you will see that the contrast between the cell size and the cell end is much larger in these natural patterns. So apparently, just to focus, to localize where the cell ends is, this simple molecule is enough, but obviously the real thing has some extra mechanisms that make it more, make the contrast higher. What is also different is the dynamic properties. So first we showed that this polarity pattern is really microtubule dependent. So if you get rid of the microtubules, and this is just a single cell, so it's not as easy to see, which is initially polarized, you put in a drug that washes out the microtubules or disassembles the microtubules, and within half an hour or so, the protein, the chimera, redistributes around the periphery. If you then wash out the drug, you re-establish the polarity pattern. So it's really microtubule dependent. And this is important to show because you have to imagine all this is happening in the background of a normal polarized cell. So you could say, your molecule is just sticking to whatever is already at the polar ends of the cells, and microtubules have nothing to do with it. But this shows, and some other controls that we did, that the microtubules really dictate here. And what is also, I'm not showing it here, interesting is that the fluctuations of the intensity are quite pronounced. So we measured the fluctuations in intensity in natural polarity patterns. So we sit at a specific spot and just ask, how does the intensity of this protein fluctuate over time? And for the natural polarity patterns, it's quite steady, whereas ours fluctuates quite a bit. So the dynamic properties of our pattern are clearly very different from the natural one, but the spatial properties are not. So we then just, and this is the one theoretical slide that I promised. I hope it will show up. It's a bit slow. We can just do a very simple modeling where we take just even a one-dimensional case where we allow particles to interact with what we call traps that come and go, just like microtubules do, and diffuse in the membrane, just to show that this diffusing and transient interactions with microtubule tips is a principle enough to get higher intensity of molecules here than here on average, and that's what you see in these curves. And these other curves are there to show that the fluctuations, as we saw also in the experiments, are actually higher at the cell pole than at the cell sites. And the difference, of course, being that at the cell pole there's constantly microtubules coming and going. I will skip that same kind of measurements for the experiments. So now that's it, as far as we've gone in the in vivo system. So I think we've shown that if you have a molecule that can diffuse in the membrane and you have microtubule tips interacting with the cell ends, that by itself is in principle enough to polarize the membrane. Of course, one point of discussion, as I already said, it's very difficult to exclude that there's not some factor that we didn't think of that is also important, because this is done in the background of a wild-type cell. But the model shows that in principle these two functionalities are enough, and you do not need anything else. So then what about this difference in dynamic properties? So what is really striking is that the intensity of these real polarity proteins is much more robust, if you want, or it doesn't respond as quickly to microtubules, and it stays a constant much more easily. So one of the reasons that this could be, and this was already known, is that actually these molecules do not sit there, it seems, as individual molecules, but they form clusters. So we have shown that if you have a molecule that's diffusing, it can be polarized, but we haven't really understood yet how can it be, when molecules have to come from the inside, from the cytoplasm, which is what happens in the real system, how can they be so precisely delivered to the first cell end first, even though we think now we understand that once they are at the membrane, that they can be kept there. So this brings me to the other approach, and I have 5 minutes, so I'll do it quickly. And which is where we didn't start with a diffusing molecule in the membrane. This is where we eventually go to, but focused instead first of, can we think of a simple mechanism that allows microtubule tips to specifically target molecules to the membrane at the cell end? Because you have to imagine all these molecules are also diffusing through the cytoplasm, and there's receptor molecules elsewhere in the cell as well, at least. There's some concentration of them. So why is the microtubule able to sort of overrule this diffusive behavior and specifically target molecules to where the cell ends are? So I'm just going to go through the steps that we thought we had to go to make this minimal in vitro system. We do not have the luxury now of an already container of the right size and shape like the in vivo cell, but we have to make it ourselves. So we have to make elongated shapes that are roughly the size and shape of yeast. We have to introduce microtubules in these chambers, and then we have to introduce Ntrackers, molecules that can interact with growing ends, and then we have to make sure somehow that these Ntrackers can interact with the boundary of our chambers. So we have to make some kind of a receptor, which is in our case just an artificial affinity for the walls of our chambers. So I'll go through this quickly. So this is just to show that, as before, we can make microfabricated chambers. And this is just a movie showing a real yeast cell with microtubules in them compared to these microfabricated chambers where microtubules also nicely align and grow. So that works. We then have to have Ntracking systems. So we borrow the same molecules that Ntrack in the fishing yeast. So the simplest one, as I already described, is MAL3. So it interacts transiently with microtubule tips, and that's why it appears to be tracking growing ends. So this is now, it's not a nice turf image. This is a spinning disk focal microscopy image, which is what we prefer for these chamber experiments, but you see still quite nicely that individual MAL3 molecules track with the ends of microtubules. So this is the one system we used, and then we compared this with the more complex system, which has been known to Ntrack in vitro. This is work that we did together with Thomas Surrey several years ago. So if you have MAL3, then you can, in combination with a kinesin motor T2 and a cargo tip 1, then these molecules will also, as a cluster, not only interact with growing ends, but also travel along microtubules to the end. So this is now an individual microtubule growing and shrinking. This is the chymograph, where you see such a complex of molecules. In this case, the cargo tip 1 is labeled and traveling at the ends. And this picture here is in no way meant to show you what is the stoichiometry or how they are arranged with each other. It is true, however, that all these molecules are dimers. They can make interaction with each other. So we think that unlike the MAL3, which just comes and goes at the tip, these complexes of molecules behave more like clusters or particles or blobs or whatever you want to call them. And you see that also nicely in this sort of anecdotal movie, where you see a microtubule that is pulled in the other direction, and the plus step meets another microtubule, and then this whole bunch of molecules at the growing tip breaks up into small clusters, which now travel along the other microtubules. So they're not behaving, we think, as individual molecules. Now we have to have affinity at the barrier. So we, as you may have noticed, the cargo or MAL3 that we use is always his-tagged, and that allows us to engineer artificial affinity to the boundary. We can use a PLL PEG with a tris nickel NTA, which we received from Jacob Pieler's lab. It's not commercially available, but this triple NTA PLL PEG, if we put it on the walls, specifically on the walls and not on the bottom of these chambers, it will have some affinity for his-tagged proteins. And you see here such a bit of rough chamber where we put his-tagged MAL3, and it binds nicely to the edges of these chambers, and it allows us also to tune the affinity, because just like on a purification column, you can put imidazole or not, and this will change the affinity of these his-tagged proteins for the boundary. So if you do not put any, everything sticks and nothing diffuses anymore. If you put a lot of imidazole, all the protein is diffusing in the chambers and nothing sticks at the wall. So we choose an intermediate concentration such that there's some affinity of these proteins for the wall, but not so high that they all stick and none of them is available to interact with microtubules anymore. So the last pieces of data that I will show you is trying to use microtubules to deliver these molecules to the boundary to which we've engineered some affinity. So this is the experiment as it's done with MAL3. So the microtubules here are not free to orient. They actually are stuck to the surface, so we're just looking at individual, what we call delivery events. You see that there's some protein in the chamber diffusing. That's why you get some of them just by diffusing, sticking to the wall. And then you have this enhanced intensity at the tips of microtubules. But if you look carefully, whenever a microtubule hits the boundary, for as long as the microtubule is there, there's an enhanced intensity of the MAL3, but as soon as the microtubule is gone, also this enhanced intensity is gone. So even though microtubules hit the boundary, after they are gone, there's no memory in the sense that MAL3 is not remaining or is preferentially binds where microtubules hit the wall. And again, this is a chymograph showing that same thing. If you do this now with the cluster, with the T2 motor, MAL3 and tip 1, and we his-tag the tip 1, it's a very different result. So again, we get some molecules sticking to the periphery just by diffusion. But if you now look at these arrowheads, you see, for example, over here, you see an individual microtubule hitting the boundary, and then after it has a catastrophe, there's still some intensity of the protein left behind. So apparently, the microtubule tip can organize these molecules at its tip in such a way that when they reach the cell boundary, that their combined affinity for the wall is higher than when proteins just arrive by diffusion, and thereby it can put a mark on the boundary after a catastrophe happens. And this is very similar to what is observed in vivo. And we think, based on these experiments, that it has really something to do with the way these proteins cluster at the tips of microtubules, and that you could just beat diffusion by using the microtubule tip as a hub to organize these molecules so that they can then will stick to, in this case, a completely artificial his-tagged wall with different kinetics than they would do by diffusion. So that's more or less where we are. We can quantify this and just show, what is the intensity at the microtubule tip when it's traveling through the chamber, and then compare it to the intensity at the boundary after the microtubule has left. This is these two signals here. And then if you do many of such experiments, you find that this is in arbitrary units, the amount of protein traveling at the microtubule tip. This is the amount that eventually stays behind at the barrier. And if you take a straight line, you say that, okay, that about 20% of what was delivered actually stays behind. And if you do the same thing with MAL3, then none of it stays behind. So really, as I already emphasized, it matters how they interact with microtubule tips. So now, of course, you would ask, okay, now you have this delivery of proteins, you have elongated chambers, so now you just put the microtubules in there freely, allow them to orient, and you can maybe make a polarized chamber. And unfortunately, that's not working yet. So one thing we forgot is when we put the microtubules free in solution, when they're not stuck to the surface, they actually sort themselves out because there's multiproteins here, and all the plus ends end up in the middle instead of at the cell ends. If you look through your air, if you take a bit of distance, you will see that most of the plus ends, most of the commons are in this area and not over here. And a yeast cell, of course, has antiparallel bundles combined with a nucleus to keep the plus ends at the outside. So this will never put any marks at the cell ends simply because the plus ends don't go there. So what we had to do is borrow from Stefan Dietz's work, where he, together with Marcel Jansson, showed that you can make actually antiparallel overlaps also in vitro. And he did it with a minus end kinesin and ACE1, which is a cross-linker molecule. We used for the same purpose dynein, and I'm just going through it quickly, but if you tune the amount of ACE1 and dynein in the correct way, you can create antiparallel microtubule bundles with a stable overlap. If you put not enough ACE1, then they simply slide and then let go. If you put too much ACE1, which breaks the motion of the dynein, then you get very strong parallel overlaps with no plus end sticking out. But somewhere in the meantime, you can get anti-parallel bundles. So this is now the situation. We have more or less polarized plus ends now, although it's not ideal yet. And what we now have to do, but haven't done yet, is functionalize the walls in these chambers and see if we can polarize them. And this chamber had no functionalized walls yet. So that experiment is still on the to-do list. There's stupid practical problems that I will not bother you with, why it's not really there yet. So that's it. We want to now combine this delivery, this putting marks on the surface with a correct distribution of the microtubule plus ends. What I will skip is that obviously in the end we want to also include, once we have delivery at the right correction in these chambers, we would like to do the same thing in droplets, elongated droplets, so that we can also include the diffusive behavior of the proteins. And just this slide shows you that we can make these elongated droplets. We can again have his tag, affinity for his tags by putting, in this case, nickel NTA lipids. And this is somewhere lower on the to-do list. So this is it. So I hope I've shown you that it's work in progress, that we can use these bottom-up reconstitution experiments to hopefully learn something about the mechanisms by which these cells can polarize, and learn from that ideally also something about how microtubules help polarity processes in other cells. And with that, I want to acknowledge the many people that have helped us over the years, that we collaborate with on different projects. And I am very particularly pleased that this polarity, we just actually got a grant from a Swiss agency to work together with Sophie Martin, who is a cell biologist expert on this problem, and to really boost this in vitro approach to eventually maybe even reconstituting the complete POM1 system in vitro. So that's It's more plans than collaborations for now. And I thank the different funding agencies and thank you for your attention.